Welcome to our channel All Room. In today's video, we are diving into fascinating world of fermentation. Especially, we will be exploring the differences between homolytic and heterolytic fermentation. So let's get started. Fermentation. Fermentation is a natural process that has been used for centuries to preserve food and create delicious flavors. It's a metabolic process where microorganisms such as bacteria or yeast convert sugar into other compounds like acids or alcohol. Now let's talk about homolytic fermentation. In this type of fermentation, microorganisms, particularly lactic acid bacteria, convert sugar into lactic as a primary end product. This process is commonly observed in the production of yogurt and certain type of cheese. During homolytic fermentation, lactic acid bacteria such as lactobacillus break down the sugar present in the milk, releasing lactic acid as a byproduct. This gives yogurt its tangy flavor and helps to preserve it by creating an acidic environment that inhibits the growth of harmful bacteria. Now let's move on the heterolytic fermentation. Unlike homolytic fermentation, this process involves the conversion of sugar into the combination of lactic acid, ethanol and carbon dioxide. Heterolytic fermentation is often associated with the production of various fermented beverages such as kefir and certain type of short beer. In addition to lactic acid bacteria, this type of fermentation also involves the participation of yeast and other microorganisms. In heterolytic fermentation, lactic acid bacteria initially convert sugar into a lactic acid, similar to homolytic fermentation. However, yeast present in the process then further metabolize the lactic acid, producing ethanol and carbon dioxide. This combination of compounds contribute to the unique flavor and effectiveness found in the certain fermented beverages. It's important to note that both homolytic fermentation and heterolytic fermentation are metabolic processes carried out by a specific group of bacteria under specific conditions. These processes play a role in various biological and industrial applications, including food production, the production of fermented beverages, and the preservation of food through fermentation. Now let's discuss the difference between homolytic fermentation and heterolytic fermentation. First, substrate. Homolytic fermentation. Single carbon source. Heterolytic fermentation. Multiple carbon sources. Second, microorganisms. Homolytic fermentation. Strictly defined species. Heterolytic fermentation. Multiple species. Third, energy sources. Homolytic fermentation. ATP. Heterolytic fermentation. ATP and reducing equivalents. Fourth, process. Homolytic fermentation. Anaerobic or aerobic. Heterolytic fermentation. Mainly anaerobic. Fifth, metabolic diversity. Homolytic fermentation. Limited metabolic pathway. Heterolytic fermentation. Broad metabolic pathway. Sixth, efficiency. Homolytic fermentation. Generally low yield. Heterolytic fermentation. Potentially higher yields. Seventh, examples. Homolytic fermentation. Ethanol production. Heterolytic fermentation. Lactic acid. It's important to note that while these characteristics generally apply to the homolytic and heterolytic fermentation, there may be exception and variation depending on the specific microorganisms and substrate used. So whether it's creamy goodness of yogurt, bulby tang of kombucha, fermentation play a vital role in creating a wide array of flavors and preserving foods. Understanding the differences between homolytic and heterolytic fermentation gives us a deeper appreciation for the science behind these age-old techniques. That's all for today's video. We hope you enjoyed the explanation of homolytic and heterolytic fermentation. If you did, give us a thumbs up and don't forget to subscribe to our channel All Room. If you want to learn a manufacturing process of yogurt and cheese, let me know in the comment section. Thanks for joining us today. We will see you in the next video.